So I'm going to talk to you about how we use kind of a host and a suite of bioinformatic tools to find phages and then engineer them to be useful in therapeutic settings. So we've seen a resurgence of phage therapy due in a large part to antibiotic and multi-antibiotic resistant bacterial infections. And in the last 3 years alone, we've had a number of high-profile cases where they've gone out and find environmental phages and use them in human treatment to treat a lot of systemic or localized infections. And the reason that phage therapy has come back is a number of different pros. They're super inexpensive, they're easy to produce. Phage cocktails overcome the single phage resistance that we've seen with antibiotics, where if you treat with one antibiotic, it becomes resistant. A bacterial cell is typically not both antibiotic and phage resistant, so you have some synergistic work there that can treat infections. And it's specific to one bacterium, which leaves your microbiome intact, allowing for increased disease clearance. Additionally, many bacteriophages have access to 2 life cycles, and the phages that have typically been used in phage therapy are those that can undergo only this lytic cycle, where once the phage genome is in the cell, it produces virions, it lyses the cell, thus killing the bacterial pathogen. However, far more bacteriophages are actually the second type, called temperate phages, which can undergo either of these 2 life cycles. And what is problematic in the phage therapy community is when the phage genome can integrate into the host chromosome and can replicate, leaving these cells resistant to future phage infection. So the question that we were asking is, how can we use this vast database of prophages to overcome this resistance mechanism? So our lab has developed 2 different algorithms that find genomic islands, such as prophages, in addition to other mobile genetic elements and integrative conjugative elements. And the first is called Islander, and it discovers these prophages only in tRNAs or tmRNAs, and they can only contain a tyrosine integrase. And what this algorithm does is it looks for this island signature pattern, where you see a tRNA gene, an integrase, and then a displaced tRNA fragment. And this was really important in finding these kinds of islands, and it also showed us that integrases are located near one end of genomic islands. So we then further developed a second tool called Comparator, which uses comparative genomics to find prophages in any genomic site, including protein-coding genes, tRNAs and tmRNAs, and intergenic space. And this is using the premise that the integrase gene is located close to one end of the island, so we take a left query from the midpoint of that integrase, and we blast it back to all reference genomes, and in our case, it's all of RefSeq. And then we find usually a broken hit. We take the right adjacent hit, and then we blast it back to our genome of interest to find the distal end of that genomic island, thus giving us precise nucleotide coordinates for this prophage. So when we use these tools together, they're complementary, and we did a small subset of about 2,000 genomes that are verified at NCBI, and we found 6,500 genomic islands, of which about 2,000 are prophages. And these 2 tools find overlapping islands as well as distinct islands. And when we break that down further into target gene types, we see these are predominantly found in protein-coding genes and tRNAs. And when we break that group of protein-coding genes down, what we find are highly conserved genes such as ribosomal proteins or metabolism genes. Additionally, we find that many bacterial pathogens have high numbers of predicted genomic islands, as shown here, where the bacteria-wide average is about 3 islands per genome, and a lot of these pathogens far surpass that. So how do we use these computational tools to influence phage therapy? So we can run Comparator and Islander on all sequenced bacterial genomes, which we have done, which is about 300,000 at NCBI right now. And we can create a database of genomic islands. And again, about a third of this will be prophages. We then can have a patient come in the clinic, and we can sequence their clinical isolate or a portion of their clinical isolate and place it phylogenetically on a tree, and then ask which of the closely related strains have high numbers of prophages. So in this case, we might select this green strain, which has 5 prophages. We then use genetic engineering tools to knock out the lysogeny genes. And the way we've done this thus far, we've used Pseudomonas aeruginosa as a proof of principle. We've created interlocking Gibson assembly long PCR fragments that delete the integrase gene, which would remove the ability to integrate into the host genome. We transform that into the target strain or the clinical strain here, and we look for phage plaques, and then we can do a number of downstream characterizations, including EM, sequencing, liquid killing assays, and wax phage therapy in waxworm Galleria melanella as well as mouse phage therapy, and then we can also use these engineered phages for patient trials. So one of the things that we really need to understand is whether or not these phages we predict are actually active prophages, or are they cryptic prophages and they're just hiding in bacterial genomes. So to do all the work that I'm going to show now, we selected 2 Pseudomonas aeruginosa strains, which had 7 prophages in total. And the first thing we did was we treated the wild-type bacterial strains with mitomycin C and used another tool we developed, Juxtaposer, which looks at next-generation sequencing reads. And if the prophage is not active, we'll see standard reads where we see the @L and the @R sites. However, if the prophage is active, we'll see these recombinant reads because the prophage will have excised, leaving a circular junction read, and it will leave the deletion junction or the bacterial closure. And when we did this, we showed that all of our prophages that we identified in these strains are active, shown here by the red line, and we can show that they're also replicating because at 2-hour time points, this circular junction far surpasses the deletion junction shown in bacterial cells. And thus far, we've engineered 6 of these bacteriophages to remove the integrase gene, and we've tested them for a number of different factors, a lot of which I won't have time to show, but I do have a poster outside, including chloroform sensitivity. We have variable plaque morphologies. We've tested them in liquid culture, and what we can show is that this is the buffer-only line, is that all of our engineered phages are these solid lines. They kill very well in liquid culture as a single phage, and we're moving to phage cocktails in the near future. We've also done some preliminary testing in the waxworm model with single phages, and it was extremely promising. We're scaling that up and again moving to phage cocktails. And we have plans to test all of our engineered phages in mouse models. And we're currently in the middle of host range studies using the CDC Pseudomonas antibiotic resistant panel to see how widespread our phages are. Because I said they're super specific to one bacterium, so engineering these phages from close relatives may or may not work. Additionally, we are looking at other ways to engineer lysogeny knockouts, so we can engineer out the entire lysogeny cassette, which contains the integrase and the repressor and additional excisase genes, or using other ways in addition to Gibson assembly, such as CRISPR-based methods or recombinering strategies. And we can also engineer in additional proteins if we want it using these methods. So I just want to leave you with, we know that bacterial strains are a rich source of prophages, and the tools that we've developed allow us to find these prophages and we know them with their nucleotide precision, so we can engineer them to remove the drawbacks that temperate phages have in use in phage therapy. With that, I'd just like to thank my postdoctoral mentor, Kelly Williams at Sandia National Labs, our collaborators at New Mexico Tech who are doing a lot of the host range studies, and our funding. And then I'll take any questions.
Thank you very much. Question, Beer.
That was a really nice talk. Is there any evidence that the, thank you. That was a really nice talk. Is there any evidence that the temperate phages in any way have an advantage as a phage therapy over the obligately lytic phages because they would be able to, for instance, overcome or persist through a population bottleneck?
So the first temperate phage therapy case was just published about a month ago. It was done on Mycobacterium abscessus, so I don't think people have actually studied head-to-head, are temperate phages better than lytic phages? Do they overcome any bottlenecks? I think the phage community still has a lot of places to go and a lot to study, and I think that's a big one.
Question?
Since you're using temperate phages that have ubiquitous integrated genomes, what's the rate of recombination back to wild type?
So we haven't looked at that yet, and that's something we probably should look at. I know that in the liquid killing studies, we do see a little bit of resistant strains come up, and whether or not that's due to reintegration, even though the integrase is removed, or it's due to some type of resistant population, that's a question to still be answered.